The floor is yours.
Okay, thank you very much, Alex, for the introduction. First, I want to thank Raoul and Christian for inviting me to give a talk in this interesting workshop. It's always a pleasure to be here at KAUST. Yeah, so today I will discuss how to use the idea of Markovian projection to do efficient filtering and importance sampling. And this will be based on joint works with the following collaborators that you see in the screen. So it's collaboration with Nadir Birashid, Raoul Tamponi, Maxime Chopin, who recently started his PhD here at KAUST, and Sophia Manka, a postdoc in RWTH Aachen, who unfortunately could not make it. So I hope that she attending the talk via Zoom. All right, so my talk will be more or less based in two parts, or split in two parts. The first one, I will focus on inverse problem, mainly a filtering problem. And there we try to design efficient finite dimensional stochastic filter to estimate conditional probability distribution of an observed state of variables given some partial observation trajectories. And there, what has been done in the literature, usually either using particle filters or solving filtering equations, lead to the curse of dimensionality. And then we try to use the idea of Markovian projection to solve the problem in a lower dimensional space to make the approach scale better with respect to the dimension. Depending on the time left in this talk, also I will look at the forward problem where we use the idea of Markovian projection to design efficient Monte Carlo estimator for relevant probabilities. And the idea there is first to formulate the problem in a way to design generic path-dependent change of measure via importance sampling, and this leads to a stochastic optimal control formulation. And when you try to solve it numerically, you face the curse of dimensionality, and there comes a Markovian projection to help efficiently deal with this challenge. All right, so first I start with some background material for stochastic reaction networks, which will be more or less the kind of processes that we deal in this talk. And I will try to motivate also where the high dimensionality comes in these problems. So our aim basically in this kind of setting is to model the time evolution of a certain population. So this population can consist of molecules, species, agents, cells, depending on the application that you have in hand, and they are connected through reaction networks. So this is a simple example that where you have two species or agents, so A and B, which are connected through three reactions. And these three reactions basically will change the counting number or the state of these two species. So if you look at the first reaction, then basically it will consume one instance from A and produce one instance from B. The second reaction is a reversible one to the first one. And then the third reaction, which is the decay reaction, will consume one instance of the reaction A. And what you observe here is that when we go to consider D types of molecules, species, or agents, then the state space will grow, number of states will grow exponentially with respect to the dimension. So here you start seeing where the dimension can hit you, and we will face the curse of dimensionality because of this. Quick overview about stochastic reaction networks. So they are continuous time discrete Markov chains, and they are driven by Poisson processes. So there were a lot of applications that used stochastic reaction networks. For instance, the spread of epidemics, one can use stochastic reaction networks. Also, for the case of transcription and translation in genomics. So here you see, for instance, where gene can switch on and off, and if it should switch it on, then it will produce mRNA, and then the mRNA will produce proteins. Okay, so these regulation systems are very important in many applications. Also, they have been used to model many types of networks like social, ecological, and logistic networks. And also, they are very useful to model biochemical reactions. And most of the examples that I will consider in this talk will be related to biochemical reactions. So for instance, this a famous example of Michaelis-Menten enzyme kinetics is a very important problem that is related to metabolism of human metabolism or many cellular activities. So this was one of the examples. All right. So let's now go to some formal formulation of stochastic reaction networks. So here, formally, we consider J reactions. So theta J here indicates the reaction rate of each reaction. And what you see here are the reactants. What you see here are the products. And so nu J basically will be related to the state change that is induced by the reaction J. So we will denote by X here the state vector of our stochastic reaction network. So each component of that state vector will be related to the counting number of the each species or molecules that you have in the system. And the dynamics of X is characterized by the pairs nu J and AJ. So as I said earlier, nu J will be related to the state change that you have in your state, given the firing of the G reaction. So the minus counterpart that you see here is for the reactants, how much you consumed, and the plus is how much you produced through the reaction J. And AJ is what is called the propensity or jump intensity function. And basically, this gives you the likelihood of the reaction J to occur. So you see here, for instance, that the probability that you observe a change in your state between time t and t plus delta t, giving the firing of the G reaction, is so it's equal to AJ evaluated at the current state. So that's why it's the likelihood of the reaction J to happen times the length of the time interval plus high order terms. Okay, so these are the two important pairs that characterize the dynamics of stochastic reaction networks. Now, to say a little bit about the propensity, so there are many models that model the propensity functions, but the most popular one is the one based on mass action kinetic principle, where the propensities here are modeled as the reaction rate, theta J, times a certain monomials, okay? So the monomials here will be dependent on the reactants that you have in your reaction. So that's why you see that it involves just nu ij minus. So you just compute this propensity based on the reactants that you have in your reaction, and they will be polynomials, okay? So the shape of these propensity function are polynomials. Now, now once we introduced the characteristics of stochastic reaction networks, we can say something about the dynamics. And this is given via the Kurtz random time change representation, which basically says that the evolution of your state vector is nothing else but the initial value, which can be deterministic or random, given a certain initial distribution, plus a certain sum over the number of reactions that happens until time t. Okay, so here Rj is the number of reactions that happens from time 0 to time t, and as you see, they are modeled as a unit rate. So they are independent unit rate Poisson processes, where the intensities here are the integration are computed as the integration from the time 0 to t of the propensity functions evaluated at the current state. Okay, so this is how we characterize the dynamics of stochastic reaction networks, and you see here illustration of the path of a certain example of a stochastic reaction network if you use this exact representation. So one example of schemes that can be built based on this characterization is the exact simulation scheme, where each time you try to sample two random variables. The first one will give you the first time for a reaction to happen. So it's exponentially random distributed and a variable. And the second one will tell you which of those reactions that are existing in the system will fire. Okay, so each time you do that until you reach the final time, capital T. However, since this is the exact scheme, as you know, in many areas, this is in most of the cases will be slow, mostly when you have some of your reactions that have high propensity, because in that case, the time for the next jump or the next reaction will be very small. Okay, so in those cases, it's not efficient to use exact simulation schemes. So that's why, okay, before that, I need to say something about the assumption that we will make about the propensities. So here, throughout the talk, we will assume that we have a uniform bound of the sum of the second moments of the propensities. So this will ensure that the system state will not grow to infinity in a finite time almost surely, okay? So how you can get it? Because you use the property of the propensity functions that are modeled like polynomials, and then this condition can guarantee that you have a certain behavior, okay? So this will be crucial for all the results that I will show later on. Now, coming back to a certain approximation that will be used in the second part of the talk. So this is the explicit Taulip scheme. And this is basically an Euler approximation of the exact representation that I showed you before. So what you do, you just need to freeze. So you have a partition of time steps that you have in your time interval. And then what you do, you just need to freeze your propensities as those time steps. And then this leads to the following discrete representation of your dynamics. So you end up with now with the Poisson random variables at each time step for each reaction with intensity that is equal to the propensity, as I said, frozen at the left point of your time step, the state evaluated at the left point of your time steps times delta t, which is the length of your time discretization. And here, okay, we need to make sure that we have positive because we are dealing with counting numbers. So one way to ensure that the discretization scheme, the bias that is induced by the discretization scheme does not violate the physical behavior of the process is just to project to zero your updates whenever you cross the bound. So again, I highlight that here, the Poisson random variables and also in the exact representation, the unit rate Poisson random processes here are independent with respect to each reaction. And when we do the Taulip, they are also independent with respect to the time steps, okay? So this is crucial. Okay, so far we have seen more or less characterization of stochastic reaction networks. Now let's motivate where the cursive dimension happens. So whenever we try to solve some problems modeled by stochastic reaction networks, these problems can be, for instance, a chemical mass equation, which is basically the forward Kolmogorov equation for discrete state space Markov chains, right? And we want to model the time evolution of the joint probability distribution. Or we want to solve the stochastic fitting problem, or we want to solve a certain stochastic optimal control problem. We will see later on that this leads to the curse of dimensionality, which means that your cost, your numerical cost, will grow exponentially with respect to the state space. So this is the case. So this is also here. So we have such kind of situation. But the nice observation is that in many problems, you can you have a quantity. Yes. So it's problem dependent. So here we treat practical cases. So you can have some cases where they are fully connected, but some cases they are not. So I will show you some examples later on. Okay, let me go to. So you have this setting, right? So you have x1 and until xd, right? And they are connected with the, so you have G reactions, right? So some of the reaction will collect some of the species or the agents that you have in the system. And I mean, for some cases, you can have maybe cases where you don't have a connection. So again, so the node J will characterize more or less the interaction because more or less it will involve the reactants with how much you consume it from the states that you have in your system, and the plus counterpart is how much you produce. So this tells you whenever they are not zero, this more or less tells you what are the states in your system that are involved in the G3 reaction. Yeah, the new, exactly, the new, the state change vector. This will give you what are the states that are affected by the G3 reaction. Does that answer your question? Okay, please interrupt me if you have any question. All right, so, okay. So what I was saying is that, okay, whenever we want to solve these kind of problems, we face the curse of dimensionality. But one needs to observe that in many cases, the quantity of interest lives in a lower dimensional space, okay? So for instance, if you want to compute a certain expectation of observable, that you just look at the indicator function, so relevant probabilities, for instance, and then you look at the marginal, maybe information of your state, so one of the components or subcomponents of your state vector, or when you do filtering. And this is one example. For instance, we have like, you have six components in your system or six species, and then you, let's say that you observe the proteins, protein 1 and protein 2, and then you are only interested in the distribution of the second mRNA or the copy number of the second mRNA. So at the end of the day, you don't care about having the information of all hidden species or molecules that you have in the system, but just you need to characterize the distribution of one of those, okay? And in those situations, you need to use that, right? You don't have to solve the problem in the full dimensional space because you will be hit by the curse of dimensionality. So you need to use such kind of structure to make more efficient numerical schemes. And this leads us to the use of Markovian projection. So the solution here is to construct a low-dimensional Markovian process that will mimic the projected process. Why Markovian? Because I think also in the first day, this was motivated in this problem that I showed you here, like stochastic optimal control or stochastic filtering or Kolmogorov forward equations. They use the Markov property, right? So either from a formulation perspective or from the numerical method that you will use, they get advantage of the Markovian or the Markov property. So we don't want to just construct a projection, but we want a Markovian projection, okay? So that we are able to keep or to use the same kind of formulation and also the same relevant numerical methods. All right, so this leads us to the Markovian projection. So coming back to the coarse random time changes representation. So as I showed you earlier, we have the following characterization of the dynamics. And let's say that we have a certain projection. This projection will depend on the problem that you are trying to solve. So you are projecting your full state to a certain low dimensional state with dimension that I denote by d bar. So what you first observe is that while X is Markovian, so the original full dimensional process is Markovian, the projection is not, okay? So this is the first observation that you can make. So again, the idea now we want to have a mimicking process in some sense that will mimic the distribution or the evolution of this projected process. And it should be Markovian. And this leads us to the following theorem. So the idea of Markovian projection, of course, is not, at least it's not new in itself. So this has been introduced with, by Giorgi around maybe the 80s or 90s. And this was for diffusion processes. So here we try to adapt it to the case of pure jump processes. So what does this theorem says is that first, okay, I assume that I have the non-explosivity condition that I have introduced earlier. And the idea is that if, so what it says is that if I define S bar, the D bar dimensional, so the stochastic process living in the lower dimensional space with dimension D bar, I define as such. So what you observe is that here now it has the same representation introduced by Kurt's random time change representation, but with the difference that I have non-homogeneous propensities here, so they are time dependent. And where these kind of time dependent propensity in the projected space are given by the condition expectation of the original propensity, given the projected process and the initial condition, then what we can show that this constructed process S bar and the projected process S will have the same conditional distribution for any time t in your time interval, given they start from the same initial condition. Okay, so this is what the Markovian projection theorem tells you. And here I'm putting two references. Why? Because you can have two ways to show that. So in the first one, we have just you use the weak error representation and then use Dynkin formula. And then you continue until you you show the that you have the same distribution, or you can work with the chemical master equation. You do some marginalization, and then you end up with a similar result. Okay, so this is how you can show such a result. Okay, so looking now at this theorem, we can make some observations, right? So the first one, as I already said, we changed a little bit the dynamics that we were dealing before in the full dimensional space. In the full dimensional space, we have the propensities. They are time homogeneous, right? So they do not depend on time. But in the projected process, it will be dependent on time. So this is the first observation that you can make. This projected propensity can be computed numerically for a certain case. So I'll show some illustration later on. But otherwise, if you want to compute them numerically, they are not given in explicit form. You can use L2 regression or Monte Carlo. And what you can observe is that you don't have to compute the projected propensities for all your reactions. Why? Because first, there will be some reactions that will not contribute to the projection. So these reactions will have, because maybe I have to say it here. What you see is that also the nodes that you were referring to before, right? Now in the projected process, so this kind of stoichiometric vectors will be given by the projection times the original propensity. So for certain reactions, they will be blind, they will be kind of killed in the projected process whenever you have the projection of the nodes equal to 0. And in those cases, you don't have to compute those projected propensities. Or if your propensity is given explicitly as a function of your projection, in that case, you can get an explicit characterization of your projected propensity. Yeah, this is what I was saying, actually. So you can lose certain interaction because when you apply the projection, certain reactions will be equal to 0 because for certain reaction, the P times nu J will be equal to 0. So I will show an example. Maybe it will make this clear. But of course, you will lose some interactions. Okay? So this is, yeah.
Talking about the example before about the that's how you but then how do you relate that to this project? I assume that's what you guys.
Yeah, this is what we go through. So you will see many examples now, and then I hope that this makes this clear to you. Otherwise, I mean, I can follow up on this.
The general step that you summarize is the number of species.
Exactly. Exactly. You do not control intentionally. So you do not do dimensional reduction with respect to the reaction because you can do that, of course. But you do it with respect to the state space, to the state vector. And then automatically, because also this representation is in a weak sense, right? So those interactions basically will contribute in second moments information that you lose, but they will not matter. And we will see later, I mean, they will matter when we go to the second application about optimal variance reduction. But first, the certain application, you don't care about that, okay? So let's see an example to make first, Christian, this is your question, okay? All right, so let's see an illustration to make this clear. So here I have an example. So this is a famous good CS model for regulated transcripts. So I have 6 species and they have 10 reactions, okay? So I will have 3 kind of sets of reactions. So the first set I denoted by, so I make it here in red. So those reactions, as you see in the reactants, they do not involve D. So here, let's say I have a certain problem that my quantity of interest only depends on the dimers, so molecule of type D, okay? So if you see the first 4 reactions, they do not, so this is not D, but this is just a composite product of D and the DNA, so it's not D itself. But in these 4 reactions, you don't have D in the reactants, which means that when you do the projection of those propensities, this automatically will give you 0. So those reactions, you don't have to compute their propensities, okay? So the red guys. Now, the last reactions, you can, so if you look at the formula of the projected propensity of that guy, you just have it explicitly because the projected process is just D. So you don't have to do any numerics to compute that propensity because it's given in explicit form with respect to your projection. Now, for the remainings, you need to do some numerical method. As I mentioned, you can do this with L2 regression, or you can do it with Monte Carlo, okay? So this is an example where you can see, like, you can have some reactions which are killed in some sense by the projection, okay? Yeah, Eric. Which one? No, this is okay. This is just a notation. So you need to see the D like as reactant, but whenever you see this dot, it's just a composed product of the DNA and D. So just it's a notation, chemical notation. So, but it's not D. Okay. So, all right. So as I said, I mean, one way to do this, for instance, to do L2 regression. So here I'm showing example of those of the reactions where you need to compute the project propensities. So this is more or less how they look like if you apply a L2 regression. So just I want to illustrate that you can do this numerically. Yes. Yeah, you do some simulation for your data, and then those data. I mean, you do not do Monte Carlo to compute the expectation, but rather to just simulation, and then the simulation will be used as a data, and then you just do training data, and then you just do your L2 regression with respect to that data. So it will involve simulation. But when I say Monte Carlo, you don't compute that expectation with Monte Carlo because you can do it also with Monte Carlo. Yes. Sure. Which product you mean? Yeah, but okay, because of the structure of the propensities, they do not contribute. You see, the propensities only care about the information in your reactants. You don't care about the products in your reaction. You see because of this new ij minus. So all the dependence, the monomial that you have there, it will be with respect to the reactants. No, you don't care about what happens for the products. Okay. All right. Yeah. So here it's just illustration of 30 paths. Now, so here this is the full dimensional state or process. So you have the different trajectory, the 6 species that are present. And here, if you apply the Markovian projection and simulate the trajectory, you observe such kind of thing. But this is maybe more interesting because, again, the statement of the Markovian projection tells you that you have the same distribution, right, of your projected process and the one that you build with the Markovian projection. And this is what you observe here. So here, if you look at the relative occurrences of the state and you compare it, if you just, so the red guy is just, you have the simulation and just compute the projection. And then you take the histogram of those. And the blue guy is just when you look at the Markovian projected component and you do a histogram, so you see the agreement also, at least empirically, that gives you confidence about. Yeah, it's in alignment with the theory. Okay, so now we had the setup for the first part for the stochastic filtering. So we introduced stochastic reaction networks. We discussed about the general concept of Markovian projection in that case, and now we want to look at the filtering problem. And this is a motivation for the filtering problem. So as I said, in practical problems, usually you don't observe fully your system. So this is a very famous example in regulation and gene switch networks. So this is one small example, but you can have symmetric examples. So you can have 2, 3, 4 blocks of those. So they increase the number of species. But in those cases, you are only able to observe the protein. Okay. And the other kind of species, like the gene, so the state of the gene, if it's activated or not, or the mRNA, that you do not observe them. Okay. So in this case, you want, let's say, for many cases, you want to get some information about the distribution or some quantitative interest about the mRNA or about your genes based on the observations or the trajectories that you have from the observed one, which is the protein in this case. So this is one example where it is relevant to look at such kind of problems. So the problem of interest is that given partial measurements of your trajectory, how to efficiently estimate the conditional probability of your unobserved or hidden state variables given what you observe. Now let's go a little bit formally. So here, if I go to the state vector, this is my full dimensional state vector, and I denote by the red guys the hidden part of my state. The blue guys are the observed parts, okay? And this is what you can observe, for instance, one trajectory of those observed processes. Now, both subcomponents will satisfy the Kurt's random time change representation. So the first one, the hidden one, it still satisfies the Kurt's random change time representation, but again, I need to separate the state with the hidden one and the observed one, and I just take into account only the stoichiometric vector or the state change vector that is contributing to the hidden state. Okay, so this is the only change. So this will be subcomponents of the stoichiometric vector that you have initially. And the same thing, you have the same representation for the observed process, with the only difference now that you only take into account the state stoichiometric vector or the state change vector contributing to that observed part. So this is the only thing. Now, formally, as I said, we want to estimate this conditional distribution, so that I denote here by pi y. So the probability of your hidden state at a certain time t, given that you have a trajectory, a continuous trajectory here, for instance, of your observed part. Okay, so this is the problem that we are trying to solve, and there are already, some theory before that makes sure that such a quantity is well boosted. In particular, you need this non-explosivity condition that I mentioned earlier. So this is crucial to make sure that this quantity is well boosted. Now, to make later on the notation clear, I need also to separate the reactions. So we separated the processes. So there will be also two kinds of reactions. What I call the observed one, that they denote by calligraphic O, and they depend on the time steps, so the jumps. So these guys basically are the reactions that introduce a change in the observed part. So this is what you observe in your problem. And between jumps, we denote by the unobserved reactions by calligraphic U, and these are the reactions which do not affect the observed part. Okay, so I need to introduce this kind of notation to make things clear. Now, before going to our approach, of course, there are a bunch of ways to do this, right? We have seen particle filters, which AJ nicely explained them. The problem with such kind of method is that when you go to high dimension, they can face some issues. For instance, they can face the sample degeneracy issue, which means that when you start with your particles, as you go in time, the effective number of particles which will contribute to your average that you are computing for the quantity of interest will be smaller and smaller, okay? So you need to get rid of those because they have a smaller weight. And then this leads to certain problem in terms of accuracy, but also robustness, because we use central limit theorem and these kind of things to make sure these quantities are well defined. And this problem, whenever you go in high dimension, starts to be more impactful. I mean, there are a lot of works mentioning such kind of issue. The second problem with particle filters, as it inherited the same problem from Monte Carlo, is when you go to estimate the tail probabilities, okay? So if you want to estimate some very small probabilities for your conditional probability, then you can get a problem with accuracy unless you increase dramatically your number of particles. Now, okay, I do not show it here, but of course you can use Kalman filters, but Kalman filters does not make sense here because we are dealing with a discrete state space first, and and it's a nonlinear system. So you can, of course, use Kalman filter, but this will introduce a certain bias in your problem. So this is another method that is not relevant in this case. And the last one is that you solve what we call filtering equation. So this filtering equation that I will introduce later will be the time evolution of the conditional distribution, and it will be given by an infinite dimensional system of coupled ODE with jumps. And these are, as I said, called the filtering equation. And since we are dealing with ODEs, it's very clear because we have the state space scales exponentially with respect to the dimension. We will see that this kind of formulation will face the cursive dimension whenever you want to solve them naively. So this is what we do. The main idea of our approach is that we will use a reduced variance particle filter to compute the projected propensities because they will depend on the observed trajectories. And then once we compute those, we will solve now the filtering equation in a lower dimensional space given by those projected propensities. So this will be the main approach that we will follow. All right, so let's say something about the filtering equation. So, okay, the first thing I need to say, I don't show you the filtering equation for the normalized probability because usually they introduce nonlinear terms and they are not friendly when you want to solve them. So we can look at the unnormalized version of that, and then we just we can take later on. I mean, we can get the quantity of interest by average and taking the sum. So the fitting equations has two kind of formulation: what occurs between the jumps and what happens other jump times. Okay, so what happens between the jumps is very similar. If you are familiar with chemical mass equation, is very similar to the chemical mass equation. So the mass, the evolution of your mass will be given by positive contribution when you transit from x minus a certain previous state to your current state, and you will deduct whatever kind of mass that you lose when you transit from your current state. Okay, so this is more or less what gives you the evolution of this equation. And at the jump times, as I said, we observe the reactions. So this is the calligraphic O that I introduced before. So we can define them explicitly because we are able to observe them. So this gives you the following formulation. So as I said earlier, you can adjust, if you solve the unnormalized version, it's straightforward later to get your quantity of interest. Yes? Someone want to ask? Okay. Okay, so once we introduce this equation, of course, I can now write them in a matrix form. So if I write them in a matrix form, this will introduce an infinite dimensional matrices, right? Because originally, we are dealing with infinite dimensional states, right? Because of the nature of the process. So your A here will be given by the terms that appear in the first equation. The matrix B will be given by the ones contributing at jump times, okay? So the first problem here is that we are dealing with an infinite dimensional system that we need to solve. And there comes the finite state projection or the filtered finite state projection. So this is first based on the finite state projection that were introduced for the sake of solving the chemical mass equation. And then it was extended later on for solving the filtering equation. And the idea basically is simple. You just you need to truncate your state space. So here I'm using a truncation parameter that is n. And in that case, now you depart from an infinite dimensional system that you had to solve earlier to a finite dimensional system. Okay, so this is the filtered finite state projection. And then there you do not have a priori estimates, but what you can do is that you can control the truncation of your state based on a certain a posteriori error estimates. Okay, this solves one part of the problem, but still you are faced with the cursive dimensionality because, again, the number of states will be scaling exponentially with respect to the dimension of your hidden process. Okay, so even doing this truncation, it does not save you from the cursive dimensionality. And now we want to call the idea of the Markovian projection that I introduced earlier. Okay, and we will, as you see, we'll adapt it a little bit to the filtering context. So again, I look at my state vector. And I introduce another color, sorry. Here, that is the orange one. Okay. So again, before we had the red and the orange were the hidden states, the blue one was the observed. But in the hidden, in the hidden state vector, you are not interested in all of them, right? You may be interested in one or two. So there are hidden species of interest, and then there are hidden species which I don't care about. Okay, so this is the usual setting that you get. So to solve this problem, you have two ways to do that, right? So the naive way, what you can do, of course, you just need to solve the full dimensional problem. You marginalize with respect to the hidden states that you don't care about. And then this gives you the conditional probability of the hidden states that you care about. Okay, so this is the naive approach. Of course, if you do it, you will face the cursive dimensionality that I spoke about earlier. But the second one is now you can try to do marginalization with respect to the process. So now you try to only look at your projected process, which is only composed of the hidden process of interest plus the observed states. Okay. Again, here the challenge is that this projected process is not Markovian. So what we need to do, the Markov projection idea. So as I said, so here you separate your processes. These are the ones that you are interested in. These are the ones that more or less you want to throw. And then the idea, we want to go to the projected space. And then we want to solve now the fitting equation in the projected dimension instead of the original dimension to avoid the curse of dimensionality. How we do this? I think depending on time, I will skip this naive approach. The naive approach, just you do the same thing I introduced earlier for the standard Markovian projection. So you see the same formulation, but what you notice, I do not condition with respect to the trajectory, with the observed trajectory. And what you expect, this will introduce a bias. But what is interesting in some numerical experiments, you see that in some cases, they give you a good accuracy. But we are aware that these kind of things will introduce a certain bias. So we need to introduce another Markovian projection, which is a filtered Markovian projection that will take into account the information generated by the observed process. And this is given by the following theorem. So it's more or less the same idea. So you construct a surrogate process, but what you observe here is that now the projected propensity are not only including a conditioning with respect to the projection at each time, but also you include the conditioning with respect to your trajectories. Okay? And this, of course, will lead to the use of particle filters to estimate those projected propensity. You cannot use anymore the Monte Carlo or A2 regression. We need to use something that takes into account the evolution of your trajectory. But again, if you construct your process as such, with the following projected propensity, you guarantee that the original projected process and the Markovian equivalent, or the Markovian projected equivalent, have the same distribution, giving a conditional on the observed trajectory. So as I said, now what you need to do, you use a theorem, you know that the Markovian projected propensity are given as such. What you can do to compute those that are not given in explicit form, you need to use particle filters and a certain extrapolation to compute those. And then what you do later on, you just apply your filtered fine state projection to the reduced system. And then, as I said earlier, this will guarantee that you have a consistency with respect to the quantity of interest that you are looking for. So before I show some numerical analysis about the approach, I just show some numerical experiments. So coming back now, we look at this stable gene switch network. So I have 2 proteins, 2 mRNAs, and 2 genes. And what I observe is only the trajectories of protein 1 and protein 2. And I'm interested in the distribution of the second mRNA. And what you see here is that I'm showing, so first, the results of the margin distribution of the mRNA. So this is the hidden trajectory. And the blue one is the FFSP when you compute the full model, where you solve it in the full dimensional model. And the red is the standard MP. The green guy is the filtered MP. And what you see, at least first qualitatively, they have a good agreement with respect to the benchmark, which is the FFSP in full dimension. And now if you look at the specific times, this is what I said earlier. For some times, you can get a good agreement between the standard MP and the filtered MP. But for certain times, you see the difference. And then it really matters that you use this filtered version of the Markovian projection. In terms of computational time, this really brings a lot, because instead of solving the original problem in 6 dimensions, which involves like around 15,000 of states, now with the projection, you are only solving your filtering equation and with respect to 31 states. And you see that you get a big gain in terms of CPU time. Another example that I go through quickly, which is the linear cascade example. So you have a connection between different species, S1 to until SD. And then, okay, the same thing here, I show you the distribution of the hidden process, which is in this case, the S5. But in this case here, it's a different behavior, because you have like a stationary behavior of of your hidden species. So good agreement between qualitatively between this method. But what I want to highlight more is, because this kind of example you can play with the dimension. So you can increase the dimension as you want, and then first you see this cursive dimension that appear for the solving the FFSP in the full dimension, and then you see that the filtered MP and the standard MP are more robust in terms of increase of computation time with respect to the dimension of your states. Now, in this plot, I compare also with respect to particle filters. So here I show the conditional probability mass function with respect to the all states for the linear cascade. And what you notice, so the blue guy is a benchmark, I would say. This is solving the FFSP in the full dimension. The green is the filtered MP, and the pink one is a particle filter. And what you will notice first is that the particle filter, when you go to the tails, starts to be suffering. So here for the particle filter, or when I use particles to compute the filtered MP, I'm using the same number of samples or the same number of particles. And you see that the particle filter will stop at a certain case, so it will not be able to compute any conditional probability. So this is the problem at the tails that I discussed earlier. However, for the filtered MP, you see still that you can get a good agreement with respect to the benchmark that you have. Okay, let me go quickly to the error analysis. So for the error analysis, we have kind of 4 kinds of error, I would say. So let's say this is our quantity of interest. This is what I compute. I compute my condition probability by solving the FFSP in low dimension. I have delta t, which is the resolution with which I solve my fitting equation. The m is the number of particles I use to compute the projected propensity. So if I do some error decomposition, this will introduce 4 errors. The first one is the model reduction error, where you go from the full dimension to the projected one. But what we know, because of the Markovian projection, the field Markovian projection, the error there is 0. Okay, so you guarantee that if you solve exactly the projected propensity, there is no error coming from the modeling point of view. But you have a projection error because you will need to use some particles to compute these projected propensities. You have a truncation error because you need to truncate your fitting equation, and then you have an ODE solver because of the delta t that you have at end. So at the end, you can show that the expectation of your L1 norm error here will be given. So this will be the contribution of the particle filter that comes when you approximate the projected propensity. This is the error coming from truncation of your fitting equation, so the FFSP, and this is the ODE solver error. So it depends on the order that you are using. So this will be the contribution of that error. Okay, so this gives you an estimate of the error that you are doing at the end of the day. So let me now ask about how much time I do have, because maybe I don't have much time left. -3. So I can quickly, in 5 minutes, go through the optimal control and then I close. Is that okay?
Yes, it's good. Please.
The chair told me it's fine. So yes. Yeah. Okay. So the first term, okay, I'm going from, so my quantity of interest. So this is if I go to the filtered Markovian projection. Okay, yeah. So the filtered Markovian projection gives me characterization of Markovian projection to make sure that the distribution of my projected process and the one I constructed as such by the Markovian projection have the same distribution. This will lead, I can show that the conditional probability that I'm interested in is the same. So here you see the thing I'm computing with respect to the original projected process, the original process, that one involves the projected process. And then you don't introduce any bias because you are you are ensuring that you have the same distribution. So in mixed sense, you do not introduce any bias. And this is what I was talking about, the modeling error.
After the projection, the loss, the interaction that comes with it, right?
The?
After the projection.
Yeah.
The loss, interaction.
In this case, yes, because I care about the weak. So here I'm computing a certain probability or expectation, right? So I don't introduce an error, but maybe the thing I cannot maybe cover is when we do this to design optimal variance reduction. And this is still kind of an open problem because we see a nice kind of a variance reduction when you do the Markovian projection, but the quality of the variance reduction that you get will be impacted by the choice of the dimension where you project to. So in that case, because you care about the second order moments in some sense, so you are talking about variances, in that case, losing interaction in the projection will impact the quality of the variance reduction that you get. In this case, you don't get the impact because you are computing expectation or probabilities, so you just conserve, the thing in weak sense. Okay. So yeah, and then I stop maybe, so I don't have to cover the slides are shared, you know, the references. So the last thing, okay, maybe I just go to the final references and then I close this thing. So if you want to learn more about, all right, yeah. So if you want to learn more about optimal control, so there are two ways we do this. So this is the paper where we do the Markovian projection, and this paper where we do it alternatively by using a learning-based approach. And for the first paper that I discussed, also you can look at the code, which Maxim has nicely put in Git, and then maybe this gives you better experience to understand more the problem. And I finished by that. Thank you.
Thank you very much. So I have one question, but maybe somebody, yeah, please.
I don't know if there's anyone online. Thanks for the nice talk, Shahib. So if you have this Markovian projection, is it sort of implicit that the memory is decaying sort of really fast?
Again?
Like if you have this Markovian projection, is it implicit that sort of the memory is decaying somehow really fast? Because otherwise,
that's a good point, because for instance, you can see it in the cascade problem. So in the cascade problem, I don't know if I think I had a plot. Yeah. So what you see, for instance, when I increase the dimension, yeah. So this is for d equal to 3. The second one for d equal to 5. What does it mean? I'm conditioning with respect to the first component. So if you look to the cascade problem, I'm conditioning with respect to the S1. So if I look at S3, S5, you expect that the dependence will kind of be less and less as you increase the length of your cascade, right? And what you see that in that case, you would expect that the standard Markovian projection will be as good as the filtered one. Because in some sense, the information that you are looking at by observing trajectory is not as important as for S5 as for S3, because you have kind of more lengthy sequence. And this is the thing that you mentioned. And then this is something that you can look in this. If you look here, for instance, when I compare the standard MP and the filtered MP, you see that they are in good agreement. And then, okay, later on, of course, the filtered MP takes over. But here you see more kind of problematic effect. Actually, the other way around. Here you see that asymptotically, that the MP starts also to be as good as the filtered MP. But here, asymptotically, the filtered MP is more kind of accurate. And this is because of that thing, if I understood you correctly, because as you go long in your sequence, so instead of the third one, you go to the hidden as your fifth one, you care less about the information that you want to record for the first species in some sense. So there is kind of also information, but this impacts if you need to do the filtering, maybe MP, or just do the standard one, and you are fine with the inconsistency in some sense.
Thank you.
Thank you very much. I have also a question, but I will ask it in pause. Somebody else, urgent questions? Okay, thank you very much for interesting talk.
Yeah, just I need to switch off this thing.
Yeah, I guess, for example, in your pocket or something, it should be fine. It's more about like working suddenly like this.
Okay. I would test it.
Support pointer. No, not this one. The pointer, the pointer. Oh, the pointer. Yes. Maybe to connect and connect. Can you move and sorry? This one, so just to connect and connect. So it will work on the connect. This one is working. Then we move here forward. Maybe this one that was working before. This is a bit slow. Okay. It's hard to control it. So wait, you can tell. Wait a bit.
Okay, the first talk of this session will be given by Anil Ben Rashed from University of Utrecht. We'll talk about free cellular wireless networks and